Now we are going to discuss about gibberellin hormone. Contents of gibberellin hormone, history of gibberellin hormone, biosynthesis of gibberellin, and the physiological effect of gibberellin, and the mechanism of action of gibberellin. First one, history of gibberellin. It is discovered by same period of vaccine hormone. In 1926, Japanese scientist identified that full seedling disease was caused by the fungus Gibberella fujikori. The pedesta was pale thin color, which is called Bacone disease. In 1935, Yabuta was isolated the active substance that hit stable and gave the name gibberellin. In 1938, Yabuta and Sumiki was isolated and identified gibberellin is a crystalline form. It is classified into two types, gibberellin A and gibberellin B, from their original preparation. In 1955, Brain et al. Was identified or obtained by single gibberellin which having acid properties, so it is called gibberellic acid. In 1960, Macmillan to be identical with gibberellin 1 earlier from Gibberella fujikori. It is now known that there are over 125 different gibberellins which have been isolated under their chemical properties. This is a structure of gibberellin hormones. Next one, physiological effect of gibberellin hormone. Seed germination, dormancy of buds, root growth, elongation of internerves, and blotting and flowering to prevention of abscisic acid hormone also helpful in the gibberellin hormone. Induced parthenography or development of seedless fruits. Parthenography means pollination. Next one, promote flowering in long-lived plants. Next one, biosynthesis of gibberellin hormone. Gibberellins are usually synthesized from methyl erythritol phosphate or mannonic acid erythrophosphate or malonate 5-pyrophosphate pathway. In higher plants in this pathway, bioactive gibberellin is produced from geranyl diphosphate or biphosphate. In the malonic acid pathway, three classes of enzymes are used to yield gibberellin from gibberellin diphosphate or bisphosphate, and also otherwise called as terpenoid synthesis. Cytochrome B450 monooxygenase 450 and 2-oxoglutarate dependent deoxygenases. There are 8 steps in the malonic acid pathway. First one, acetate is converted into acetyl-CoA. Acetyl-CoA is converted into acetyl-CoA with the help of the enzyme or transferred by the enzyme acetyl-CoA acetyl transferase. Acetyl-CoA is converted into beta-hydroxy beta-methyl butyl-CoA, and it is acetyl-CoA is converted into CoA-SH with the help of the enzyme Sjctnc-J.John synthase. Sjctnc-J.John is converted into malonic acid, and the 2-NADPH2 is converted into 2-NADP, and the removal of 2-NADP molecules is particularly removal of 2-hydrogen molecule reduction by the enzyme Sjctnc-J.John reductase. Malonic acid is converted into malonic 5-pyrophosphate or malonic acid 5-pyrophosphate, and 2-ADP is converted into 2-ADP with the help of the enzyme malonic acid kinase or pyrophosphate malonate kinase or phosphomalonate kinase. Malonic acid pyrophosphate is converted into isobutyryl pyrophosphate and removal of carbon dioxide and water molecule with the help of the enzyme diphosphomalonate decarboxylase. Isobutyryl pyrophosphate is converted into dimethyl pyrophosphate with the help of the enzyme isobutyryl pyrophosphate isomerase. Isobutyryl pyrophosphate which having 5 carbon atoms and dimethyl pyrophosphate which having 5 carbon atoms. The isobutyryl pyrophosphate is combined to dimethyl pyrophosphate to form geranyl pyrophosphate. So 5 plus 5, 10 carbon atoms. So a geranyl pyrophosphate is which having 10 carbon atoms. It is also otherwise called as monoterpenes. The geranyl pyrophosphate is combined to 1 isobutyryl pyrophosphate to form farnesyl pyrophosphate. So 10 plus 5, total 15 carbon atoms. It is otherwise called as sesquiterpenes. Farnesyl pyrophosphate is combined to 1 isobutyryl pyrophosphate to form geranyl pyrophosphate which having 20 carbon atoms. It is otherwise called as diterpenes. Geranyl pyrophosphate inhibited by MO1618 and phosphon D to form couplet pyrophosphate and removal of one carbon molecule. The couplet pyrophosphate is converted into N-current and inhibited by phosphon D. N-current is converted into Gibberellin 12 aldehyde and inhibited by paclobutrazole. The Gibberellin 12 aldehyde is converted into Gibberellin 12 and Gibberellin 53. The Gibberellin 12 and Gibberellin 53 is combined to form all other gibberellins. Next one, mechanism of action. First one, promotion of stem elongation. The gibberellin is inactive regulators of the inactive to negative regulators of the SPY, GAI and RGA. SPY means spindle mutants, RGA means gibberellin inducers and RGA means reverse mutants or mutants of the gibberellin hormones. The SPY enhances the actions of inducer gibberellin and reverse mutants. So in absence of the gibberellin inducer and reverse mutants act to suppress the growth to form mRNA transcription leading to growth. So finally the stem for elongation of the root plant. Next one, mobilization of endosperm food reserves. The gibberellin combines with a receptor on the outer surface of plasma membrane of alluron layer cell. The gibberellin receptor complex interacts with a heterotrimeric G protein and initiates two separate signal transduction pathways. First one, a calmodulin or calcium independent signal transduction pathway, and another one, a calcium dependent signal transduction pathway. The calcium signal transduction pathway which involves cyclic GMP as a signaling intermediate leading to the expression of alpha amylase gene. A calcium dependent signal transduction pathway which involves calcium binding protein, calmodulin and protein kinase signaling intermediates leading to the stimulation of alpha amylase and other hydrolytic enzymes from cells of alluron layer into the cell or endosperm of starch degradation. The gibberellin signaling intermediate is activated. The activated gibberellin signaling intermediate goes into the nucleus and binds with delta repressor proteins. For example, inducer gibberellin, reverse mutants and also spindle mutants. The delta repressor and SPY are degraded or inactivated or switch off. Due to inactivation or degradation of these repressors, the gibberellin MIB and other genes are switched on. The gibberellin MIB mRNA goes into the translation and gibberellin MIB protein transcription factors are synthesized. The gibberellin MIB protein enters into the nucleus and binds with the promoter gene for alpha amylase and other hydrolytic enzymes. Alpha amylase gene and other genes that encode other hydrolytic enzymes are transcribed. Alpha amylase mRNA moves out from nucleus to rough endoplasmic reticulum in cytoplasm for translation process, and alpha amylase protein are synthesized. Alpha amylase protein are secreted via Golgi bodies. Finally, the alpha amylase is converted into degradation of starch in endosperm. Thank you.